Hello, my name is Richard Crick, and I'll be presenting on cell death. Cell death terminates normal cellular functions, including respiration, metabolism, growth, and proliferation. Cell death can be non programmed, for example, as a result of accidental injury or trauma, or programmed. Types of programmed cell death include apoptosis, autophagy, necrosis, necroptopsis, and pyrotopsis. Cell death is completely normal and it goes on all the time in your body. In fact, death of cells is essential for healthy development and homeostasis. However, not all cell death is beneficial. Beneficial cell death is one that is carried out in a controlled manner. Wherever a single cell malfunctions or becomes diseased, then apoptosis occurs. This is a highly regulated breakdown of that cell coupled with the production of a new one, thus maintaining a relatively constant process of life and death. Some causes for apoptosis to occur include cells infected with a virus, cells impaired beyond repair, cells in a stressful state, as known as starvation, cell DNA damaged by oxidants or other agents. Apoptosis is an organized way of removing a non functional cell so that it can no longer drain nutrients nor spreads infection. On the other hand, necrosis means death of a group of cells in the same area caused by lack of blood. Necrosis caused demise in ungoverned and irreversible. There are various categories of necrosis, named for their location or type of tissue destroyed. For instance, fat necrosis, skin necrosis, tumor necrosis, intestinal necrosis, acute tubular necrosis, renal papillary necrosis, osteonecrosis, which is also known as avascular necrosis. Causes of cellular necrosis can be attributed to a number of health conditions or outside instigators like injury, burns, shock, hernia, cancer, fibrosis, hypoxia, poisons, frostbite, radiation, abscess, infarction, vasculitis, embolism, chemicals, thrombosis, hypotension, inflammation, corticosteroids, and venomous snake bites. For an example of one would be a brown recluse infection. Making a simple comparison between apoptosis to necrosis, some differences are one, that apoptosis is not fatal and necrosis can be. Two, apoptosis is controlled cell death, necrosis is not. Three, necrosis typically causes inflammation, whereas apoptosis does not cause inflammation. Four, apoptosis often provides a beneficial effect. Necrosis is usually detrimental. Five, necrosis causes death to a group of cells in an area, while apoptosis is sensational of an individual cell. Once necrosis causes a substantial number of cells to die, the, the consequential health condition is known as gangrene. Apoptosis is the original term introduced by Kerr to define a type of cell death with specific morphological features. Apoptosis is not a synonym of programmed cell death or cap space activation. Autophagic cell death defines cell death occurring within autophagy. Though it may mi- though it may be misleading, suggests though it may be a misleading suggestive form of death occurring by autophagy, as this process often promotes cell survival. Cornified envelope formation or keratinization is the specific of the skin to create a barrier function. Although apoptosis can be induced by injury in the basal epidermal layer, cornification is exclusive of the upper layers. Necrosis identifies in a negative fashion cell death lacking the features of apoptosis or autophagy. Note that, that necrosis can occur in a regulated fashion involving precise sequence of signals. Irreversible cell injury. Mitochondria swell, lysosomes swell, damage to plasma membrane and lysosome membranes leads to enzyme leakage. Um, Excuse that. As an as a usually healthy form of cell's life cycle, apoptosis rarely demands any form of medical treatment. But untreated necrosis can lead to series of injury and or death. Both apoptosis and necrosis can be seen as part of a spectrum of shared biochemical events that both result in some form of cellular death. Apoptosis, or programmed cell death, causes cells to shrink, develop blebs on the cell membrane, undergo degradation of genetic and protein materials in the nucleus, and have their mitochondria break down, thus releasing cytochrome, each wrapped into their own membrane, which other chemicals release freely. These chemicals lead macrophages, cell-eating bodies, to find, eliminate, and dead cells and their fragments. This message is triggered by a phospholipid normally inserted in a cell's membrane, and the macrophages, in turn, release cytokines that inhibit inflammatory responses. In contrast, necrotic cells swell or may form vascules on their surface, with interior structures either distending or shrinking rapidly, destroying the cell's processes and chemical structures. The first role of apoptosis is during the intran- intrauterine development. It helps to sculpture organ shape and carve out the interdigital webs of the fingers and toes. Apopto- Mechanisms are important deterrents of fetal abnormalities. Experiments have shown that wild type P53 mouse embryos will readily abort after radiation induced ter- teratogenesis, whereas P53 null embryos will not. Both the nervous system and the immune system arise through overproduction of cells, followed by the apoptotic cell death of that which is fail of that those that fail to establish synap- functional connections or productive antigen specificities. This is a picture of the morphology of apoptosis. The cell would shrink, then collapse into crescents around the envelope. Then the envelope would then bulge and bleb. And after the blebbing would occur, it would then increase, and the cell would finally break into an apoptotic body. Autophagy allows the orderly degradation and recycling of cellular components. In macrophagology, targeted cytoplasmic constituents are isolated from the rest of the cell within the double membrane vesicle known as autophagosome. The autophagosome eventually fuses with lysosomes and the contents are degraded and recycled. Two additional forms of autophagy are also commonly described: mitotic autophagy and chaperone-mediated autophagy. In disease, autophagy has been seen as an adaptive response to stress, which promotes survival, whereas in other cases, it death and morbidity. In the extreme case of starvation, the breakdown of cellular components promotes cellular survival by maintaining cellular energy levels. The intrinsic apoptotic pathway is characterized by permeabilization of the mitochondria and release of cytochrome C in the cytoplasm. Cytochrome C then forms a multi-protein complex known as apoptosome and initiates activation of the capside through capase 9. Thank you, and this concludes my cell death presentation.